Hi, I'm Davide Confalonieri and I'm the founder of Lab Propose. And today we will be talking about how new medicaments, new therapies are developed. So if this is something that is useful for you, if you ever wonder what is happening in the lab, what are people doing, what are the big pharma doing, maybe you are a researcher working in animal health in the university and want to understand more what are the next steps, then stay tuned because we have a quite big journey ahead of us. Okay, so everything starts basically from the university. That will be the first step. That's the basic research where people trying to understand, okay, what are the bases of a particular disease? What is it that is, causing this disease? What is the molecular pattern or pathway? That it's underlying a particular disease, and basically, can I recognize anything that makes any sense in this disease? And here we're still in the realm of the so-called target identification. So in the first step, people, researchers, also in pharma, they want to understand what is the pharmacological target that I'm going to target. What is the one protein pathway or whatever that if I target it, then I block the disease forever, or at least I reduce the amount of disease. And this is really, really dependent from the kind of disease. If I have obviously a tumor, then I would have a completely different kind of target comparing to a metabolic disease or maybe a cardiovascular or something like this. And also, it's not just the kind of molecules that I'm targeting, but also the final effect that would be important. What is happening if I actually remove, block, or whatever this particular target. And so everything starts like a little bit like this. And the moment at the beginning, it's quite general. There's a lot of literature around a particular target from universities, from different research institutions, projects that are published and stuff. And that's the starting point of a lot of pharma research. Now, obviously, there's a lot of pharma companies that are bringing this in-house, especially the big ones. They are really establishing kind of basic research centers and kind of positioning themselves a little bit like similar to academia. But in most of the cases, it's actually coming from academia. And then the first step from the pharma perspective would be doing a literature review, looking what are the possible targets that are available, and then the real big step for the pharmas, for the developers of the drugs and therapies is the so-called target validation. So they need to understand, okay, there's a lot of things published on this particular target. Is this all anecdotal information, or is there something true? Is this something that we can use? How often is this particular target expressed? This is expressed in all different animals, or is this just in particular species? Also there, if I'm going to a particular species, this expressed in all particular races, or is this just for one particular one? There's obviously a lot of variability. If you're going at human, then most of the time you have a handful of ethnicities. So if you're going into the animal world, that it's completely absurd the kind of variability that you have. So depending on the kind of disease, you can have a lot of variability to deal with. And then the issue is, okay, first of all, do I find it? Second, is this a target that I can use? Is this a target that I can target pharmacologically or whatever else? What happens, if I remove the target? What happens if I inhibit it, or if I downregulate it? Is this something that is giving me any particular issue? Is this target pharmacologically relevant, basically? Is this something that I can build a pipeline on? It would probably cost me hundreds of millions, and the next, cost me the next 10 or 15 years, probably. It's a little bit less in the development of medicaments for animals, but still it's quite a long process in general. So that would be the first starting point, and there are different approaches to this. It depends a little bit on the philosophy of the company. It depends a little bit where they're coming from, what is the kind of background, and what is known also about the particular diseases. There are some diseases that are already known in the market. I mean, in the clinic, from the perspective of the pharma, obviously in the market, they are known in the clinic. There are maybe some solutions already available, and they are working for one reason or the other. So a lot of things are coming maybe from like botanical perspective. So there are plant-based medicaments or solutions that are already used, and they're working in one way or the other. So the question is, why? Why is it that they are working? Maybe there's some active ingredient in that it's working for one reason or the other, and people that would start from this kind of approach, they kind of have the key in their hand, and they are trying to search the right door that needs to be opened with this key. And so this all is the kind of it's a phenotypical approach, because what I do is I take the cells as they are. I take the tissue or whatever, and then I look at the phenotype. I look at the behavior of these cells. I'm talking about the primary cells here, isolated directly from the animal. Primary cells, tissues. Sometimes they are cryopreserved tissue, fresh ones. Sometimes companies start with the isolated cells, and now there are the kind of targets that are established, that have been validated. They are much more fine, much more not fine, as like more. The difference between the different targets are much more subtle. So you need to have some tools to look at these also at a single cell level. So there are new kind of solutions. Lab4Pose is one of the companies that's offering this. For example, dissociated tumor cells, where you're taking tissue, we're taking the tumor tissue and the normal associated tissue, and then you're taking these into single cells, and they can then be used to basically analyze the tissue at a single cell level, and then basically clustering the different cells that are more or less tumors that are in a particular stage or the other. Yeah, but this is the phenotypic approach. I start with the starting tissue, and then I target it with a particular drug. And then I see what happens. And with a particular drug, a particular solution, and so. And the idea is, number one, that I can deconvolute my target. That's the name, target deconvolution, so that I can understand basically what the target is, how is this working, and if I can target it in other ways that is a little bit more, other ways that are probably a little bit more, effective or safer, also for the animal, without a lot of the side effects. And on the other side, we have the a little bit more, I would not say classical approach, because actually it's probably the one that developed kind of later, but it's establishing a lot, and there's a lot of controversy and discussions in the pharma world about which one of the two is best. But the second approach is the target based. So I know already the target from literature, and then my aim is finding out the medicament, the candidate basically that is working the best. And both kind of approaches, they aim at coming at a point where you have where you basically have a target, something that you know that you can use to develop a therapy, and that you can study well, that you can either the structure is known, or you can in a way produce the target, have it crystallized, have it send it to some labs, and they give you back the structure. Nowadays a lot of the techniques that are going into this direction. They also use a lot of AI, artificial intelligence. There's a lot of programs that are coming up, which is also an interesting topic, because some of these solutions are limited in kind of licenses, but I will not go down this this road. But basically, the aim at the end of the target validation is understanding what your target is, how does it look like, and this will be the base for building all the remaining part afterwards. And I'd say for today's videos, we will stop here, and we will have the next step of the development of drugs in the next video. Then see you soon.